Welcome everybody. My name is Donagh Berry. I'm coming to you from a foggy foothills of the Knockmeal Downs in County Waterford, and I am your chair today. So I'd like to welcome you to the latest session of Teagasc's Research Insights webinar series, where we're focusing on the challenges and opportunities of the agri-food sector, with a particular focus on the latest research from Teagasc. So we have a really exciting lineup for you today. We have 3 world-class geneticists who have come through the Teagasc Walsh Fellowship PhD scheme, and each of those geneticists has spent significant time abroad honing their skills to bring it back to Ireland. So what the 3 experts today are, they're going to take you on a bit of a journey in animal breeding terms, starting off with the basics of animal breeding and how this can be improved by using DNA, or what we call genomic technologies. And then finally, we're going to finish up with how can we collapse all these vast quantities of information that we're generating both in animal breeding and outside of animal breeding into what we call decision support tools, or tools that help value creation decisions to be made. Please note that the webinar has been recorded, and there is a Q&A function at the bottom of the Zoom where you can ask your questions. I will see all of your questions and I will relay them to the speakers. Also, if we don't get time to answer those questions, we will put the answers to those questions up on the Teagasc website afterwards. So without further ado, I'd like to welcome firstly Professor Sinéad McParland, who is going to talk to us about the basics of animal breeding. Take it away, Sinéad.
Thanks, Donagh. Good morning, everybody. So as Donagh mentioned, we are going to be talking about advances in animal breeding this morning, but my job this morning is to go back to the basics of genetic evaluations and explain some terms to you, genetic gain, what it is, how we measure it, and hopefully give you a foundation for understanding Áine and Fíona's talks later on this morning, where they will be discussing advances. Most of my references this morning will be to our livestock populations of cattle and sheep, but I want to draw on some experiences of poultry and the thoroughbred industries, because these really show extreme genetic gain, and I think it'll help to visualize some of the concepts I want to talk about. So this image here, some of you may have seen it already. It's an image that was taken in 2005 of 2 distinct broiler lines of chicken. The one on the left was a line from the 1950s. This was an experimental line in the US that was left untouched. So no selection pressure at all for genetic gain. These birds were allowed randomly mate, and this was the result. The bird on the right is from a commercial line of broilers intensively selected for genetic gain. The bird on the right is 400% bigger than the bird on the left. So this is genetic gain in action, and I'll be referring back to this slide later on in the talk. So genetic gain in its simplest sense is improvement we see every generation. Now this improvement can be negative if we're talking about a trait like fertility, for example, where we're talking about days between calvings, days between lambing. The reduction can also be called genetic gain. And this little plot here is what we call a normal distribution. And most of the traits that we include in our genetic evaluations follow this normal distribution, where you have lots of average animals for the trait, a small number of really bad animals for whatever trait we're looking at, and then a small number of really good animals for the trait we're looking at. By selecting the best animals in the population to be the parents of the next generation, we achieve genetic gain. And then continuing this on generation after generation is where we see genetic gain accumulating and improving throughout our population. So how do we go about this? How do we achieve this genetic gain, and how do we identify these parents of the next generation? Now, before everybody turns off the computers and runs away, an equation in one of the first slides of this talk, I don't want to scare you with this. This is what we call the breeder's equation, and I'm going to go through this really slowly this morning. And as a disclaimer, this is coming up in Áine's talk next as well. So, and to be honest, I think most of you will know most of these terms already and are applying them already anyway. So change in genetic gain that we see every year is a function of how intensely we select the parents of the next generation, how accurately we can identify these parents as the next generation, how much variation is in the population that we're looking at, and how long all of this takes. So firstly, intensity of selection. Now, I've already mentioned that most of our traits look like this, where you've had lots of average animals for the trait and animals that are better for the trait. And by using the animals that are better for the trait as the parents of the next generation, you achieve genetic gain. Now, better than can simply mean anywhere to the right of the average animals. However, if we select the very, very best animals, this is intensely selecting, we will achieve more genetic gain than just merely selecting better than average. The next term I want to explain is accuracy of selection. So it's all well and good saying, pick the best animals, but how do we most accurately identify who are the best animals? And there's two aspects that really influence this accurate selection. The first is the heritability of whatever trait we're looking at. Heritability refers to how much variation we see in the population is due to just genetics alone. And when I think about heritability, I like to think about ourselves, our own families, think about your parents, think about your children if you have them, and traits like eye color, hair color, these things that are passing down through your family, and how much of this is just due to genetics alone. So to explain this, we look at different types, the different traits that we have in our populations, the different traits we're interested in can have different degrees of heritability, or different amounts of variation is explained just by genetics. So if we think of height, for example, now I know height isn't something we really look at or measure too much in our livestock populations, but I use this as an example because it is one of the most highly heritable traits, whether it's in livestock or it's in humans. About 80% of the variation we see in the population is just because of genetics. So if you have tall parents, you're more than likely going to be tall. For this reason, if we're collecting data in the national herd to estimate genetic potential of this bull to produce tall daughters, we don't need a huge amount of information. A small amount of daughters recorded on the ground will tell us the genetic potential with fairly good accuracy of this bull. If we take a trait that's more moderately heritable, like our production traits, whether it's meat production or milk production, about 30 to 40% of the variation we see in our population is just because of genetics. But as producers, you know that there's a lot more to it than that. And for example, diet will have a big influence on how much milk or the carcass value of an animal. Because of this, we need to know more information on the daughters of this bull. We need to see these daughters represented in different types of production systems across the country, different types of diets. And then if we take a trait like fertility or health, for example, these are what we class as lowly heritable traits. About 5 or maybe 10% of variation is explained just by genetics. We need a lot more information on these sorts of traits to get an accurate picture of the genetic potential of a bull for these traits. We need to see these animals represented across much more diverse conditions. So heritability is one aspect influencing the accuracy of selection. The other then is the type and amount of data that is recorded. For example, is a trait recorded on an animal itself, or is it recorded on its ascendants or descendants, on its family? So using milk production as an example, and I use milk production as an example because it's an easy one to visualize. Every lactation, an animal leaves a performance record. And this information feeds into her genetic evaluation. But we don't use just that to tell us her genetic potential. In fact, every time one of her parents performs, we include that. Every time one of her sisters, whether it's a full sister who she shares half of her genes in common with, or a half sister, these records will feed into this cow's genetic evaluation, as will her daughter's performance or her granddaughters and great-granddaughters. So what we're doing here is we're building up a picture of the potential genetics of this animal through all of these different sources of information coming from right across different herds. To do this, it is absolutely essential that we are having data recorded from lots of different environments, different management systems, different types of animal, where there's different breeds, ages, stages of life, for example. And that's where producers like yourselves, it's so important that we're getting this data recorded as accurately as possible to have as accurate as possible genetic evaluations. The third aspect influencing genetic gain is how much variation is in the population which we are selecting from. So it's all well and good to say pick the best in the population, but what if the very best in the population aren't much better than the very worst in the population for a trait or than the average? You can still achieve genetic gain. So if you're selecting intensely, you're selecting the very best animals, even if they're not much better than the population average, by going down this route, you are still going to achieve genetic gain. It just won't be as much as if you have a large variation in your population. So for this, I will talk about the thoroughbred industry. Now, there's much dispute over whether thoroughbred horses are getting faster or not. There's recent studies to show that they are getting faster. Whether they are or whether they're not, it is not disputed that there's not much genetic variation left in the population. Because of this, the rate of genetic gain is quite slow. Now, this isn't the only reason. There are more reasons for this, but lack of genetic variation in the population will limit the amount of genetic gain that we can see. So finally, I'm going to talk about generation interval in terms of its impact on genetic gain. Generation interval, you see, is lying underneath the line, which means the smaller this value, the more genetic gain, or the faster the rate of genetic gain. Generation interval refers to the average age of parents when all of their offspring are born. So the shorter this is, the better. I've already spoken back at the start of this talk about poultry and how great genetic gain they saw over a 50-year period. And I've just reflected on the thoroughbred industry and how little genetic gain they see. The average generation interval of poultry when offspring are born is just under 1 year using traditional evaluations. The average age of thoroughbreds is 12, which means in a 12-year period, poultry will have undergone 12 rounds of intense genetic selection, where thoroughbreds have undergone 1. Obviously, this is going to impact on their genetic gain. So, achieving genetic gain should be relatively simple, shouldn't it? You just pick the best animals for a trait and use those as the parents of the next generation. But I've also shown you that management influences. Diet, for example, can have a big influence on how these traits are shown in the population. So how do we know when we're picking the best animals? We are actually picking the animals with the best genetic potential, and not just the animals that are being managed to the best. And this is the whole crux of genetic evaluations. It's disentangling this management effect from the genetic potential so that we get the true genetic potential of our animals. I've shown you this slide already where we have this bull. He has a very good representation across the country. He has daughters in lots of different herds. They're on different types of diets. They're on different types of management systems. And this is very, very good, but this is not the end of the story when we're talking about genetic evaluations. These daughters are not evaluated on their own potential alone. Rather, we do what is called contemporary comparisons. We're evaluating his daughters relative to all the rest of the daughters, all of the rest of the cows in the herd, daughters of different bulls. So animals are not evaluated alone, but rather how they're doing relative to the others in the herd, which is removing the bias then of management from these animals' genetic potential. This gives us then figures for every animal in every herd. And you can see up in the top here, up in the north of the country, the black and white cows doing very, very well relative to the brown animal. Down the south, these two daughters from different sires are performing in contrast. And these sorts of comparisons across all the different animals in the herd, across all the different herds in the country, that give us unbiased estimates of genetic potential for animals, which we use in our genetic evaluations. What you see, however, is this is an example of a page from a beef catalogue, and you see these figures coming up. I'm not going to talk about the first lot. These are the overall index figures, and Fíona is going to be covering this really, really well later on this morning. I just wanted to draw your attention to these index values. These are the expected performance of the progeny. And then the reliability is how confident we are in the value given for performance. And this comes back to what I've already spoken about this morning, how heritable the trait is, how much information we have, the type of information we have, the degree of relationship between the animals that this information was recorded on. So to conclude, the role of traditional genetic evaluations is to disentangle management from genetics so we get the true genetic potential of animals. By accurately and intensely identifying the very best animals at as young an age as possible, as parents of the next generation, we will maximize genetic gain. But to do this, data recording is absolutely vital, as accurately as possible and across as many production systems as possible. I'd like to thank you for your attention and hand back to Donagh.
That's brilliant, Sinead. Thanks very much. Really interesting there. 80% of the differences amongst us as humans in our height is actually due to genetics. Really, really interesting. So there will be opportunities to ask Sinead questions, and I see some questions already coming in on the Q&A. Just stick them into the Q&A in the bottom, and once we're finished the whole session, we'll have 15 minutes for discussion. So thanks again, Sinead. And moving on now to Dr. Áine O'Brien, who is our sheep geneticist and also actually is a sheep farmer, a County Waterford sheep farmer. So take it away, Áine.
Very much. So yeah, Sinead went through a lot of kind of what traditional genetic evaluations look like, and I'm just going to go through some DNA-based breeding. So DNA-based breeding, or genomics is a word that we throw around a lot these days, is probably one of the biggest kind of advancements that has been made within the last couple of years. So genomics came in first for dairy in Ireland, and that came in 2009. It's since come in for beef cattle in 2015, and it's in for sheep now as well since last year. So before I jump to all of the information on genomics, I'm just going to go through some of the basic parts that contribute to it so that we're all on the same page and that we know exactly where it comes from. So the first thing I'm going to talk about is DNA. So we all know that DNA is the same in every single cell of your body, and it doesn't change throughout your life. And that's how criminals can be caught if they leave DNA at a crime scene, whether that be 2 weeks later, five years later, or 20 years later. And we know that we get 50% of our DNA from our mothers and 50% of DNA from our fathers. So the image on the right of your screen there is a story that I came across around this time last year. And I think it shows the kind of the power of DNA in a cow sphere. So this story came from Wales. So what happened over there was that the cattle from neighbour 1's farm broke into neighbour 2's field. Neighbour 1 went and got most of his heifers back, so they're all back, but one of his heifers was missing. Neighbour 2 denied ever seeing that heifer. He said that, no, I didn't see her, haven't come across her at all. But what actually happened was that neighbour 2 retagged that heifer, pretended it was his own, and carried on with his business. Eventually, neighbour 1 saw his lost heifer in the field across the way, and he reported it to the police. And this ended up in a court case, and it ended up with neighbour 2 shelling out lots of court fees and paying £4,000 in damages. So it's just a kind of a real life example of the power of DNA and being careful about where your cattle break into. So DNA is the building blocks of all genes. And we know that it's got 4 different nucleotides, so it's 4 different letters. So those are adenine, cytosine, guanine, and thymine. We don't need to know exactly what they are, but we do need to know that it's the combination of these letters that tell us a lot of the differences at different places within our DNA. So whether it's an A or a T or a G or a C, these tell us the different genotypes of the different places within the DNA. So if we then go on to, as geneticists, we talk about SNPs an awful lot. So we throw around this word SNPs, but I suppose what does that actually mean? So 99.9% of our DNA is identical. So 99.9% of my DNA is identical to yours. But most of our differences are in the form of SNPs. So I've got 2 different animals here, and they've each got a strand of DNA. We know that DNA occurs in double strands, so in pairs, but for the sake of this example, I've just identified one strand for each animal. And you can see that it's a lot of these different letters. So the A's, G's, T's, and C's are throughout the strands for both animals, and they look pretty similar. But if you actually were to look closely, you can see that there's just one little difference. And this is what we call a SNP. It's a single nucleotide polymorphism. So single just means that there's one. Nucleotide is what we call all of these letters, these A's, G's, C's, and T's. And polymorphism is just a fancy word for a change. So these are the things that we want to look for when we're trying to identify the differences between animals. So identify those animals that are better at producing milk or those that are worse, and for lots of different traits. So then how do we measure these SNPs? How do we get this information? So I'm just going to bring you quickly through the genotyping process. It's not something that you need to be particularly aware of, but it is just interesting to see how we're able to get all of this data. So here, for example, a sheep is tagged. So you can do this for lambs at birth or for calves at birth. They're tagged with a DNA tag, which is quite similar to a BVD tag for cattle. And this tag encapsulates a little piece of the tissue from the ear, and it's encapsulated in a tube with some preservative. So then that tag that sample is sent off to the lab, and two of the most common labs that we use are Neogen and Weatherby's. Most of the cattle and sheep genotyping here in Ireland occurs in Weatherby's, which have its labs up in Kildare. So the next thing then is that the sample undergoes some preparation. So the DNA is extracted from the ear tissue and is prepared to go on to these things that we call SNP chips or genotyping panels. So you can see an example of those genotyping panels there at the bottom right of your screen. And if you haven't seen them before, they're just these pretty thin glass little segments, and they're about 2.5 inches tall and maybe 1 inch to 1.5 inches wide. And that's how we're able to estimate our SNPs. So then how do we use those genotyping panels? So this is another example of a genotyping panel, and you can see within it there's 24 different segments. And what we do is that we load the DNA of one animal per section of this. So for example, we just, after the DNA is prepared, it's put onto these genotyping panels, and then within each of these sections, there's about 50,000 SNPs. So for both cattle and sheep, our normal genotyping panel contains about 50,000 SNPs, which are contained in each of these little segments. So the DNA from the animal binds in specific locations, and then there's just some processing that goes on. So the DNA is stained with fluorescent dye, and it's prepared to go into one of our genotyping machines. So this is an example of an eye scan. It's an Illumina piece of equipment, which is just a brand of genotyping panel. And you can see that there are 4 genotyping panels prepared now to go into that tray. So what happens within this machine is that it blasts different color fluorescent light onto these genotyping panels, and then the colors of light that are emitted from the panels tells us our genotypes. So that's how we're able to identify some of those differences within the DNA of animals. So after we've gotten our results back, so we are after genotyping those animals now, all of those genotype results go back to our central databases. So ICBF for cattle and Sheep Ireland for sheep. These are inputted into the national databases, and then there's quite a bit that goes on. So genomic evaluations you'll be familiar with, things like parentage and mating advice on the cattle side. But we don't really release kind of all of those raw results. So each of those results are then distributed to farmers in the form of decision support tools. And that's something that Fíona is going to discuss in detail later on. And that's how the farmer is able to see it. So for example, with an EBI or from the terminal and replacement indexes. So then what is genomic selection? And genomic selection is probably one of the main things that we are familiar with from DNA, certainly. So here we have a population of dairy cattle, and this is what we're going to call our reference population. So each of these animals have got a genotype, so their sample has been sent to the lab and we've gotten the results back. but they also have lots and lots of measurements. So these are the measurements that we call phenotypes. So things like milk yield, calving difficulties, calving interval, pregnancy to first service, and everything like that. So what we do from this reference population, which we have an awful lot of information on, is that we are able to identify the associations between the performance of that reference population and the SNPs that they have genotyped. So here, for example, if you look at SNP1, codes for +5 for milk and -0.6 for fertility. So we know that milk and fertility are antagonistic traits, so they're never really going to be going in the same direction. And we were able to get these associations for about 50,000 SNPs, as I mentioned earlier, which is the common density for genotyping platforms here in Ireland. So from this, then, we're able to develop some prediction equations. And these prediction equations we hold on to because we're going to use them in just a second. So then we have an animal that's, we'll say, a calf that is born down at the bottom left of the screen. That calf was tagged at birth and it's got its genotype back within 3 weeks. So we already have lots of information on that calf, despite not having any phenotypic data. So we don't have any measurements on it. We obviously can't measure milk yield or, weaning weight or something like that on a newborn calf. But what we do is we estimate the genomic relationship or the DNA relationship between the calf and that reference population that we have loads and loads of information on. And we're then able to use those prediction equations that we developed earlier. And then we're able to estimate the direct genomic value for that calf. So we're able to get a measurement on that calf despite not having any, real life data. So, things like milk yield or weight gain or anything like that. So that direct genomic value is then blended with our pedigree, and then we're getting our genomic estimated breeding value. And they're the values that Sinead showed you earlier, so the +5, -6, that we're able to then give to farmers. So that's the genomic evaluations, but we have there's a huge amount of uses for DNA that we can bring into it. So we have things like breed composition, we can look at inbreeding, we can look at monitoring major genes, monitoring some minor defects. And one of those big things is parentage. And that's one that I'm just going to show you a quick example of here. So here, when we're determining parentage, you can see that we have a sire and a calf here, or a bull and a calf. And as I mentioned, our DNA strands occur in pairs. So we know again that 50% of our DNA comes from our mother and 50% of our DNA comes from our father. And what we do when we're trying to estimate parentage or trying to determine if parentage is correct or incorrect is that we just compare the strands of DNA between the bull and the calf. So here we can see that it worked out lovely and that this bull is the correct sire. Obviously, there's occurrences where it doesn't work out correctly, so you have parentage mismatches, but because we have such a bank of DNA collected now and so many genotypes collected, we're actually able to suggest another possible parent or another possible sire for that animal if there is a parentage mismatch. So Sinead showed you this breeder's equation earlier, and I'm just going to show a quick example of it when it comes to the impact of parentage. So we know that annual genetic gain is a function of selection intensity, the accuracy of selection, the genetic standard deviation or the genetic variance, and the generation interval. But we also know that parentage can have an effect on the genetic gain. So this is just a graph, and I promise I'll simplify it in just a second. So here we're looking at where we have a parentage error rate of 10%, which is in between the error rates for dairy cattle and sheep. Dairy cattle will be a little bit lower and sheep will be a little bit higher. So this means that we've got 10% parentage mismatches here. And if we look at this graph, so we've got the number of progeny per parent along the bottom, and we've got the proportional reduction in genetic gain along the side. And I'm just going to simplify it here. So we're just going to focus on this blue line that codes for the heritability of about 6%, which is kind of like lameness in sheep. So that means that 6% of the differences we see in the trait are just due to genetics. But you can also see that where we have parentage errors, genetic gain really does suffer, especially if you've got few progeny. So it's really, really important that we try and minimize our parentage errors such that we can, promote genetic gain as much as we can, so that we're not trying to, kind of kick ourselves in the foot by accidentally reducing genetic gain. And it's easy to have a parentage error. So say, for example, in sheep, you could have 2 ewes lamb down in a pen and a lamb switch or something like that. You can also have, if you were to AI a cow in the morning and a bull broke in or something like that, parentage error rates are easy to come across, but we should really do everything we can to minimize them. So just in a quick summary slide, so I've just brought you through some of the information on DNA and how we measure it through genotypes, how we estimate those differences in the form of SNPs. We just had a quick look at some of the genomic selection and how it works. And ultimately, the most important thing for genomic selection is our reference population. So that's that population of animals that have got both genotypes and lots of measurements on them. But we now know that genomic evaluations aren't the only thing that we can get from DNA, so from this genotype. There's a whole host of lots of different things that we can also gain from those genotypes, and that we can also put into use as well. So that's my final slide, and I'll hand you back over to Donagh.
That's excellent, Áine. Thanks a million. That's a really exciting slide, that one that was just up, about all the potential stuff that you can get out of DNA. So Sinead and Áine have both spoke about creating all this vast amount of data, and we hear a lot about it in the media, etc. All this datafication of agriculture. So what Dr. Fíona Dunne is going to talk about now is really the hard bit: is how do we collapse all that massive quantities of data into tools that farmers and producers across the entire industry can use to make better decisions. So Fíona, take it away.
Hi everyone. So as Donagh said, my name's Fíona Dunne. I'm a postdoctoral researcher here at the VistaMilk Science Foundation Ireland Research Centre based in Moore Park. And I'm going to talk to you about decision support in breeding decisions. So before we kick things off, I want you to think about how many decisions do you think you make daily? And I suppose when we think about this, we realize we actually make an awful lot of decisions. So whether or not I have tea or coffee in the morning, but these decisions have different levels of consequences. So if I have coffee in the morning, it won't really impact my day too much, but deciding on what AI bull to introduce into my herd will have a huge impact on the future performance of the herd. And when these decisions have larger consequences, I suppose it's nice to have a decision support tool there to provide you with data-driven metrics that will provide you with confidence and facilitate the decision-making process. And that's what I'm going to be talking to you today about. So we're going to go through what decision support tools are, the key drivers behind these decision support tools. So Sinead and Áine introduced this as well, and also the application of these decision support tools. So starting off, what are they? Well, a common definition of decision support tools is that they're a computer-based technology that facilitate the decision-making process. So as Sinead and Áine introduced, data is really important when it comes to decision support tools, and they underpin these tools. So we collect information like phenotypic or performance information, such as carcass or calving data. We also include the likes of what Áine was talking about, so genomic information, DNA information on the animals. And all of this information gets fed into a computer-based technology where algorithms are then ran. And we also run our genetic evaluations and estimate the genetic merit of different animals. And we analyze the data, and then this information is produced in a comprehensive report, like the Irish Cattle Breeding Federation's Herd Plus. And then all of this information is split into various user-friendly decision support tools. And these are specific to each sector. So we have the likes of the Economic Breeding Index in the dairy sector, the Terminal Index in the beef sector, and also the Replacement Index as an example for the sheep sector. But I suppose we can take a step back and really think outside the box when it comes to decision support tools, that they don't have to be computer-based. So they're also software-based, likes of email and text message alerts on when to, say, apply fertilizer onto the land. Also within mobile applications. Then we also have the likes of maps as well. So if you're surveying your land, a paper map is still a sort of decision support tool. And then finally, we have farm advisors as well. So these are individuals that will research new information and new advances, interpret the data, and then provide you with guidance when implementing these new technologies on the farm. So all of these are sort of a decision support tool as well. But I suppose we're going to be focusing on computer-based technology today. So this is a graph here of all the different breeding decision support tools available within the sheep, beef, and dairy sector. So the top two rows are the sheep terminal and replacement index. Then we have the middle two, which is the beef terminal index and the beef replacement index. And then the bottom two rows here are the economic breeding index and the dairy beef index. And this graph just really demonstrates that it's not a one size fits all. All the traits included in these decision support tools reflect the industry and provide support that's relevant for that industry. So we're going to talk about the key drivers of and what's behind these decision support tools. So it ultimately comes down to money, profit. It's all based around profit. Sinead mentioned how we identify important traits that we will include in these decision support tools. The likes of that they have to be heritable. There has to be variance within the trait as well. And then we run our genetic evaluations. So we're able to estimate the genetic value of these animals for each trait within a decision support tool. But we also run these economic models. So these take into account the cost associated with the traits and also the income. And then we're able to estimate economic values for each trait. So in order to come up with an index value, we simply multiply the genetic merit for trait 1 times the economic value for trait 1. And we really just sum up all of these values and we end up with a single figure index value. And I suppose this is where Sinead introduced this idea of providing this single figure index value as a quick snapshot of the value that the animal can bring to your herd. And I'll go through some of these index listed here, the replacement terminal and dairy beef index, in a second. So ultimately, what we want to do with these decision support tools is improve the average performance of our herds. So for instance, as Sinead mentioned, we want to shift the performance in the desirable direction. So here's the average performance for year 1. And we want to get to this point, which is improving performance in the following year or with the next generation. And we do this in 2 different ways. So the first is selecting superior animals to breed from. And that's where the likes of these breeding decision support tools come into play. And the second way is then we can also select inferior animals to cull from the herd. And these are the sort of management decision support tools that are available. So I'm going to go through some of the breeding decision support tools that are available and that were developed here in Teagasc in collaboration with the Irish Cattle Breeding Federation. So we have, within the dairy sector, we have the likes of the Economic Breeding Index. And as I said before, all of these decision support tools are driven by profit and are underpinned by the genetic merit of these animals. So the objective of the Economic Breeding Index is to provide you a value that shows the profit per lactation. The Dairy Beef Index then, the DBI, shows you how profitable an animal's progeny will be for slaughter. So for instance, the animal that was in the report that Sinead and I showed, it had an index value of €97. So this was how profitable that animal's progeny will be for slaughter. Then within the beef sector and the sheep sector, I'm going to combine these because they have similar objectives. And really the replacement index within both the beef and sheep sector shows how profitable an animal's daughter will be per calving within the beef sector or per lambing within the sheep sector. Then we have the likes of the terminal index. So this shows you how profitable an animal's progeny will be for slaughter. And the common factor across all of the breeding decision support tools is they are showing how profitable an animal will be, or an animal's progeny. So we've spoken about applying these breeding decision support tools to select superior parents to be the parents of the next generation. But we can also select inferior animals that you want to cull from the herd. So for instance, selecting animals that are on the left-hand side of the dashed line. Once we cull these, then we shift the performance of the herd in a desirable direction. So one of the decision support tools that provides this guidance and facilitates these kind of decisions within the dairy sector is the cow's own worth, or the COW. So again, this is underpinned by profit and the genetic merit of these animals, and it estimates the remaining lifetime profit potential of dairy cows. We've also done research here in Teagasc in collaboration with Irish Cattle Breeding Federation again, and developed the beef female's profit potential. So this is similar to the cow's own worth, but it's relevant for the beef sector. And it estimates the remaining lifetime profit potential of beef females. So not only beef cows, but also beef heifers. This way you can look at their BFPP value and see if they're worth more to be kept as replacements, or should the females heifers be finished for beef production. So there's all these management decision support tools to aid and facilitate the decision-making process. So I've talked about selecting superior animals to breed from, selecting inferior animals to cull from. And I suppose the huge advantage to these decision support tools is that they're very complementary of each other. So for instance, within the dairy sector, you can use the EBI to select your superior females to breed these so that you're producing replacements for the herd. Then you can go on to use the dairy beef index to breed your remaining females within the herd, as the progeny will probably be going for slaughter. And not only this, but you can use the cow's own worth index then to identify inferior animals from within your herd that you want to cull. Now, what happens if you have a report? You can see the value. The suggestion is indicating, or the value of the animal is indicating that you, for instance, should cull them. And what if you don't agree? Well, I think this is the beauty of these decision support tools, that it's a single figure, but behind this single figure is a very complex amount of traits that are feeding into this single figure value. And if you want, you can dive as far into the information as possible. So for instance, perhaps there's a trait that's dragging down the animal that you haven't considered. And I suppose the nice thing is that you can get as complex with the decision support tool as you want. So just to tie up the last three presentations, Sinead went through talking about how we select these decision support tools, we select the data and how important quality data is feeding into these decision support tools. Áine introduced the fact that we have all this genomic information and the power of including DNA-based technologies within these decision support tools. So ultimately, all of this information gets fed in, then we're able to analyze the data and provide you with a decision support tool that aids in the decision-making process. Ultimately, the user decides on what direction they want to go, but it's about providing confidence to the user and providing data-driven metrics behind each decision. So in summary then, what we've gone through today is what are decision support tools. So they're data-driven tools that guide you when making decisions on farm. They provide you with confidence in the decision. The key driver behind all of these decision support tools, be it breeding decisions or management decisions, is always profit, and they're underpinned by the animal's genetic merit. And then we can apply these on our farms by identifying superior animals to breed from, but also identifying inferior animals to cull from. and I suppose making more genetic gain by combining them both together. And they are complementary with each other. So I've put my email address here. If you think of any questions along the way, feel free to drop me an email, or I can provide you with the papers that back up all of this information. So thanks, Donagh.
Okay, excellent. Thanks, Fíona. And we can get Áine and Sinead's videos back on. So just to remind you again, there's a few questions coming in throughout on the Q&A is at the very, very bottom. We've got 15 minutes to fire some questions. Again, I said that these three world-class geneticists on the whole area of animal breeding, be it dairy cows, beef, sheep, and I'm sure they'll chance their arm at some chickens and pigs as well. It is really great to have seen how science, cutting edge science, has the pipelines that exist all the way through industry with ICBF and Sheep Ireland to actually deliver these decision support tools at the end. But just the first question there is to Sinead. It's on the whole issue of selection intensity. So Sinead, you show that in your breeder's equation. But if you do select more intensely, the question is will this increase the reliability? And I'll just add on to that question. If it does increase the reliability, what are the mitigating options or scenarios or strategies we have to reduce an accumulation of inbreeding?
So you're asking the impact of increasing selection intensity on inbreeding?
Yep.
Yeah. You're right. And it is something we have to be really, really conscious of. What we find is the best animals in a population do tend to be more related to one another. So it is something we have to be really, really careful of when we're selecting our animals. But it is something that we're very conscious of at an industry level. So I work with some of the breeding companies in identifying animals for use in the breeding program. And one of the things we'll do is making sure that there is genetic diversity there. Ye as breeders will aim to reduce inbreeding in your herd. This is what, I mean, most people are conscious of inbreeding and we're trying to do it. We as an industry have to make sure that there is genetic diversity there for ye to select more diverse animals from. So we have to make sure that at the top of the lists of whether it's an active bull list or whatever you're selecting from, there is diversity there for ye to be selecting from. It's something that we worried about, I suppose, with the impact of genomics was how much greater intensity selection we were going to see because of this much faster rate in genetic gain. But actually, genomics has helped us because we can now see the actual relationship between animals, not the predicted relationships, which is what we had been using. And throughout my talk, I talked about predicted relationships. It was the average relationship that we see between parents and offspring and between brothers and sisters. At a genomic level, these can really, really vary. So we can use this information to much better identify suitable animals that are less related.
Okay, thanks, Sinead. Next one is a quick one for you, Áine. What's the cost of genotyping? And maybe I'll add to that one as well, has it reduced or do you think it's going to reduce any further? So cattle and sheep.
Yeah, so to answer your first question, the cost of genotyping for cattle is €22 and for sheep, it's still up around €25. So it has reduced dramatically. So I can speak from personal experience on the sheep side. So I did a lot of my PhD on sheep, and when we started genotyping sheep in 2015, the cost of a genotyping panel with 50,000 SNPs was €65, and that is now gone down to €25. So it has reduced in just a few short years.
Okay. After flying around the place. And there was a question there for you, Fíona, as well as going in order. Something about, is there new decision support tools being developed as part of VistaMilk? So things for trading calves or something like that. From Frank Amara asked that.
Yeah, so within VistaMilk, we are coming up with decision support tools like the Beef Zone Worth. So this uses the genetic merit of calves, and we're able to predict the eventual carcass revenue of these calves. Because when ultimately you're going into a livestock auction, you tend to be making decisions on how much you're willing to spend on these animals, really on how they look. but we're using genetic information to back up these decisions and estimate how much they will be, I suppose, their carcass revenue at the end of the day when you are slaughtering them. So to give you an indication of how much they will be worth when you're buying them.
Okay. If you want to, you can add the names to the people in front of it. This is one, I know, because it's an order, we'll send it to Sinead. So it's about single trait selection, Sinead. Is it happening? And the example is milk and fertility. They're negatively correlated. So how can you actually improve the two of them if they're antagonistically correlated with each other? Or do people still single trait select?
The majority of farmers these days now will be using the indexes that Fíona talked about. The reason being that they work, and they work better than single trait selection because you are simultaneously selecting for all traits that impact on profit together. So we know there is an antagonistic correlation between milk and fertility. There's an antagonistic correlation between production and fertility in general. So that's the reason for the advent of these indexes in the first place, was to prevent this single trait selection going too far ahead in one trait and leaving another trait behind, or even worse, deteriorating another trait, which is what we saw all around the world in terms of our milk and fertility. The indexes that Fíona talked about take all of these relationships into account, so that it allows us, and we can see, and all our genetic trends are showing us, that we are improving in all of the traits, even if they're antagonistically correlated at the one time. So we strongly encourage the use of indexes, single trait selection, if there is a particular goal that you are going for in your own herd and you are conscious and aware of the impact this could have on other traits.
Okay, and a follow on from that one, is there, for you as well, Sinead, is there, or Fíona can take it, or is there traits that are currently being evaluated at the moment, genetically evaluated, that are not included in the indexes, Sinead?
There are. So if you go on to Herd Plus, or if I'll take the dairy and the dairy population first, there are additional traits. There's TB and liver fluke. The type traits, for example, are all evaluated at every national genetic evaluation run. These traits are evaluated. Figures are available for every animal. These do not all feed into the index. For sheep, however, all of the traits that are evaluated are included in the index.
Okay.
I can chime in there as well that we're doing research here in Teagasc as well with the likes of age at slaughter. So there's large improvements here that can be made, because if we can select animals that reach a finishing point faster, not only are you getting the same revenue for the animal, but you're also feeding them less, and they have less consequences then on the climate too.
That's an excellent point, Fíona, and it shows just the benefit of the sustainable approach that animal breeding actually gives, because it's cumulative and permanent, and you build on it on year. Just following on some from Sinead's point, Margaret Good has a question. It'll be on TB on cattle, but I'm going to send it to Áine on the sheep. Áine, how much impact does health and longevity have on the profitability estimates? You can use your sheep example or cattle.
Yeah, so with using the sheep example, so we are currently working on the longevity side of sheep, and that hasn't been introduced to the indexes yet. But we have done a good bit of work on the health side. So we've got two health traits included in the sheep indexes now. So they are lameness and dagginess. And at the moment, they have got quite a low emphasis in the index. So they wouldn't contribute a lot to the profitability output. But what we can see is that although those traits individually themselves mightn't explicitly contribute to the profitability, something like lameness is going to also be captured within, some of your weight traits. So like weaning weight or 40-day weight, and you can see that, where there's lame animals, they're going to have a lower weight. So at the moment within the sheep indexes, the emphasis on health traits in the terminal index is about 1%, and in the replacement index is about 2%. And because they're quite recently introduced, we can then kind of look forward in the future to be placing more emphasis on those traits, but also to be looking at bringing in other traits into the index. So something like mastitis in ewes.
Yeah, that was actually going to be my follow-on question. Is there any other traits that are of particular interest, like Frank from Mara here talking about methane? Is there any bit of work going on methane in sheep or beef? Or?
Yeah, so from the sheep perspective, there's quite a bit going on with regard to methane. So in Athenry, we've got some pack chambers, and these are chambers that we're able to put sheep into for an hour individually, and that we're able to get estimations of methane from them. So Edel has got a trial going on there at the moment where they are looking at animals at different stages of their lives. So as hoggets, as ewe lambs, and as they go through different lambings. So yeah, there's quite a bit going on with methane at the moment.
Okay,
and I'd like to come in there as well. Oh, sorry.
No, but
Can I come in as well? Just in terms of methane work in the dairy world, I suppose one of the big impacts that we have, or one of the big limits we have to breeding for any trait is data, and not having enough data, as I spoke about in my talk, where we need data on all the different, the good, the bad, and the ugly right across all different types of herds. And this is a big limiting factor when you think of something like methane, because you as producers, you don't have a way of measuring methane in your herds at the moment. But this is something I'm working on at the moment, is using routine milk recording for dairy cows to predict the methane output. And there is initial results there that look good, not in Ireland, but using some of our data. And if this were to work, then this is a way that we could start to record methane across the national herd to give us a more accurate picture and potentially use this then for breeding purposes.
okay, and while you're still talking, Sinead, Dennis Hurley has a tricky one. So we've seen 20 years of gains of milk solids in dairying. Dennis is asking, is that sustainable? Can we see another 20 years going? I guess it's probably an inbreeding related question.
Yeah, there's different ways of looking at this, Dennis. I suppose if we look at our breeder's equation, how much variability is there left? How intensely can we select these animals? Over the last 20 years, we've had excellent tools introduced, like genomics, that has absolutely revolutionized the dairy industry. Will something else like this come along that can help us to even more intensely and accurately select animals? Genomics also reduced the generation interval significantly, which also accelerated our genetic gain. Can I see 20 years ahead as something like that going to come in the pipeline? I don't know. And then as Donagh mentioned, there's the whole inbreeding question. How much more intensely can we select without starting to impact on genetic gain or on inbreeding? And we know that inbreeding impacts on genetic gain then, because the associated effects of inbreeding, the long-term effects of inbreeding, are reduced genetic gain.
Okay, great.
That answer we just get out of it.
What's the typical range and the average for the different indexes, do you know? So say for when a calf is born, what's the reliability of the calf versus when it's genomically selected versus when it's proven?
So the reliability,
Yes, for Fíona.
Yeah. So the reliability of the animal is a quarter of the reliability of the sire plus a quarter of the reliability of the dam. So reliability figures start off at about a range from between 30% to 50% usually.
Okay. And Áine, on the sheep side, what kind of reliabilities, or you use accuracy, of course, which is the square root of reliability. What kind of values do you get there?
So we're seeing similar, so it's still going to be a quarter of the sire and a quarter of the dam. But we have seen an increase, kind of a 12-point increase with genomics, so it has made an impact there.
Excellent. Fíona, back to you again. You kind of asked it earlier on, these decision support tools, how do you see these evolving?
Well, I suppose they're really dynamic in nature. So with the inclusion and as research goes on into improving different traits or the inclusion of different traits, the decision support tools themselves are dynamic so that these new traits can be incorporated into the decision support tool. So for instance, we saw this with the likes of the EBI, started off with milk and fertility, then in 2005, we introduced the calving traits, and then 2006 introducing the health traits. So I suppose just this does reflect that the fact that these decision support tools are dynamic to the inclusion of upcoming traits that we're researching here.
Great. Look, we've time for one more. Just squeeze this one in on you. Any idea what the parentage errors in the beef, sheep, and dairy populations are? Do you know?
Yeah. So in dairy, we're looking at between kind of 6 to 7%. That's a little bit lower than what it is in beef. So beef is about 11 to 12%, and then sheep is a little bit higher at kind of 13 to 14%. But I can again just speak from the sheep perspective that when we started genotyping a couple of years ago, our parentage errors were up around 16 to 17%. So we are slowly reducing them, and the more animals that we're genotyping, the further we're reducing those parentage errors as well.
Excellent, excellent. Look, it's 10:30, everybody. So firstly, from my perspective, thank you all for attending, and I'd especially like to thank our three excellent speakers. I told you they were taking us on a journey. I think they did a really good job on doing that, and really it kind of sets down the gauntlet for the next group of people. This is going to be on this next research seminar is on the 18th of March. Again, three excellent speakers, experts in their fields, and these new reproductive technologies are real closely related really to an animal breeding perspective. So with that, thank you all for attending, and thanks for questions. I know there's a few that we didn't finally get to answer, but they'll be put up on the website, and the whole recording is also there. So thank you everybody again.